Thus, each of these gametes would also contain a C1. Then this winds up just being a two-cell Punnett square, because we have a single gamete produced by one parent, A2B1C2, and two gametes produced by the other parent, A1B1C1 and A1B2C1. So filling down. And then we fill across. And so when we're looking for the trihybrid genotype, that's only present in that cell, which is 50% or half fraction of the offspring. And as I said, this is a trihybrid genotype. There are two ways to solve this problem. The first one is, again, to remember that for genotypes that aren't given, we'll just assume that they're homozygous for the one allele. So that would add those genotypes to those individuals. And then we need to identify the haplotypes of their gametes. So that first parent is going to have four different types. And I know that because they're heterozygous for two genes, so they're going to make four different combinations. There's going to be A1s with B1s, A1s with B2s, A2s with B1s, and A2s with B2s. But because they're homozygous at C, all four of those haplotypes just contain C1. So those are all four distinct haplotypes of the first parent. And the second parent is homozygous at two loci, B and C. So really, that parent is only going to produce two types of gametes. Ones with A1 have to be B1 and C1, and then those that have A2, but will always be combined with B1 and C1 alleles. So the second parent only produces two types of gametes, not four. So what fraction of offspring will have that genotype? We'll need to draw out the Punnett square, and it will be a 2x4 Punnett square. With the blue haplotypes down this side. And I'm just omitting the semicolons. And the red haplotypes across the top. And then we're going to look for A1, A1, B1, B1, C1, C1. However, before I fill this in, I'm going to point out that the second parent does not have any C1 alleles. So there's no way to produce an offspring that is homozygous C1. This parent will definitely provide a gamete that contains a C1, but this one cannot. This parent doesn't contain a C1 allele. So without having to do all this work of writing out the gamete haplotypes and all that, we could have just started at the beginning and asked, is it even possible, not what fraction of offspring, is it possible to produce offspring of this genotype with these starting genotypes.